So people are playing with chemicals to enhance the human capacity. They're also experimenting now with how do you add a device to the mammalian brain to give it an extra sensory ability. You may not want to detect infrared. You might want to have a room temperature detector of radiation, depending on what your job is in life. So when you think about it, the possibility now is there to develop different kinds of devices. They could be perhaps used either by intelligence people or by people in the military to have an extra ability to be able to see through walls, to see heartbeats. We used to play with the 18 gigahertz microwave detectors where we could pick up heartbeats through anything but solid steel and water, but that could easily be a human who can see the unique heartbeat that's behind the wall over there that's thermal insensitive. So it doesn't have to be IR. It can be a number of things. Anything that you can co-opt is theoretically now possible to adapt to human brain functioning. All you'd have to learn is the code. You'd have to train with it. It might not be natural at first. You might not understand the signal you're getting, but you can add to human brain function. You can also use it to intercept signals. The experiment that was just released this last month, as I said, demonstrated that people could transfer knowledge from one human to another. And I commented to a couple of my colleagues and I said, I think right now the most direct application of that is going to be either covert communication or running drones. The set of experiments, I didn't have videos to show you, but there have been a series that have shown you can connect the human brain to a rat and control its motor movement and its tail. So you can have non-human animal drones. You can have the human brain probably run a regular drone at this point, but running a non-human drone, something like a cockroach or a rat, would it be awesome. And now if you were watching the Olympics and you see the coordinated maze of drones, the software is now really readily available where you could have hordes of little creatures that can gain access to facilities or move around in different places, all run by a person sitting in a booth. It's no more technically challenging once you do that than figuring out the logistics of how you're going to send your signal somewhere else in the world and how to protect that signal. But that's now. That's not in the future. So as you begin to think what's in 5 years, the interfaces are going to become more delicate, more refined, and as transcranial magnetic stimulation, it's a rather crude instrument right now. It creates a field that excites just hordes of neurons. But as they refine the technology so you can get a better point specificity to the neurons you actually want to activate, you should be able to do this without penetrating the skull. Either someone could wear a cap, and in fact that's how the latest brain-to-brain communication in humans was done. It was done without surgery and actually signaling via some stimulation to the retina and the brain decoding it. Although the person consciously didn't know what the code was, the brain did. So that I would recommend people becoming aware of that from the human drone technology standpoint. The second field that people may or may not be aware of, I always tell my students, I said I wasn't around when they developed atomic weapons, but Dr. Ventnor's work is, my view, the equivalent of the development of nuclear weapons when you realize that he created life in a cell back in 2010. I don't know if people are familiar with his work, but this technology paired with something called CRISPR, which is like an editing software for genes, makes a number of things immediately available. What he did is he programmed yeast cells to produce anything he wanted. They can produce perfume, they can produce petroleum, they can produce any peptide, anything we program the DNA to do, and it's in the living cell. Right? So in medicine, the goal in medicine now is to be able to do designer medicine and therapy. If we can design a cell to get into your body and release the right product for you, you won't be losing half the drugs you take through your liver when you swallow a pill and it gets digested. These can be inserted into you through the hypospray needles, almost like Dr. McCoy on Star Trek, giving you the hyperspray. It just blasts now plasmids into your squamous cells. But Ventnor was able to do that and has the patent on the technology. But you can engineer anything. You can engineer a unique thing that would only kill one person in the world. It's how it's done. You put in a specific gene slicing, you program what you like, you put it in the cell, and it can reproduce and make as much as you like. For those of you who don't know, your DNA is usually all wrapped up in tight little coils. And so what you're doing is when they create plasmids and put them into cells, it sends a signal and tells which portion of the DNA should unwrap, unfold, and produce a product. Now, this is the future of medicine. When you look at this technology in medicine and say, this is going to be done to help people, right? We want to be able to give them medicines. We actually want to correct for genetic deficits. If a kid's born with a genetic anomaly, with the CRISPR technology, the feeling is we can create the portion of the gene they're missing and go have it spliced back in. And that may help a child, either if it's in utero development or once they're older, to have the missing substance actively produced. What would you do with this if you were in security and intelligence? Well, you can do a number of things. You could decide, if you make this gene, we know that certain people in the world who function at very high altitudes very, very well do it because they had a special mutation in their genome that we don't have because we didn't grow up in the Himalayas. But they can function at very high altitudes. Could you give this to people who are going to have to do warfighting in high altitudes and they don't require extra support? Their body makes a much more efficient use and can work under conditions of lower oxygen than the rest of us. You start letting your mind wander. Can it also produce a substance that lets you function longer underwater without oxygen? But these are run by certain mutations in genes, and with CRISPR, we have the ability to actually make these and see what happens when we give them to animals, non-human or human animals, that don't have it naturally. You could have the Forrest Gump gene. You guys have been tracking. There's a gene that just makes you stronger. I would say that most of this technology is probably going to be employed by a state and not non-state actors because it's quite technical. But I say that with a caveat. When we studied Shinrikyo, if people remember, they had both uranium mines and regular laboratories where they experimented on both animals and had a whole series of laboratory experiments to develop the different kinds of gases that they wanted. Their goal was to actually mine uranium and probably come up with their own version of a nuclear weapon. But they recruited scientists, PhD level folks, and their goal was to be the rightful people running the country of Japan. But we can't assume that just because they're non-state actors, they will not make use of some technology around this. Related to this is an idea called DREADs. These are designer receptors that can be remotely controlled. So think about it for a moment. You can create a designer receptor, you can create a cell, you can put it somewhere in the body, and you can remotely activate it when the brain's exposed to the right signal. Using this technology, people have been able to transfer memories from one fruit fly to another by signaling through a light stimulus into the retina. Right now, in most animals, it's done by putting a substance into their body that will actually activate the neuron in the way that you want it. So you have the capacity to create any product. As long as you know the DNA sequence, you can insert it into a living system, and you can remotely control it. So in medicine, we think about how we do that to help people, how we do to repair deficits. Other people are going to think about how do they do it to expand possibilities. And one of the challenges that we have is that when you create a cell and you put it in somebody's body, you have to figure out where you want it. What if you want it in their brain? Right? If you want it in their brain and you can't figure out, you don't want to do surgery to plant it in their brain, if I want a product produced in your brain that may affect the way you think, the way you act, one route to that is through stem cells. If you're a quick brush up on your biology, stem cells are cells, they call them God cells. They can turn into anything. They hold the potential, unlike other cells in your body, to become anything you want them to become. And they can go find their home in the body and park there and do the work that you'd like them to do. You can infuse them, and they will find their way into the brain. So once you know that the technology is there to edit, splice, and program a cell, and the technology currently exists to administer it to somebody and have it go park anywhere you program it to go park, proliferate, and do its function, you can have things activated in other people's brains. So if you take these 3 key points, hopefully you can see it opens up a number of both alarming and exciting possibilities. You can have the timed release of information on demand. Hopefully, when I mentioned the word CRISPR and word editing and creating molecules with CRISPR and playing with DNA, some of you thought encryption and encoding. So DNA encryption, there were, I think, 8 articles published by China in the course of 3 years, in the last 3 years. And it's quite important. The coding system, DNA steganography, I'll just say the short story on this is people have figured out how to hide imagery in the DNA of bacteria, and when you phosphoresce the bacteria, you can discover the information, or you can have those are just to remind me, you can have the information reproduced in a string form as a form of a protein. Dr. Church up at Harvard has shown quite well that you can store a lot of information in 1 gram of DNA. It's essentially, that many iPads in 1 gram at room temperature. No supercooling required. DNA is highly stable. It's been around on the planet a very long time. So between CRISPR, the storage capacity, and programming cells, the new way to hide information is going to be in DNA. The commercial application is going to be a bit like on Star Trek years ago. Why would you have a digital system when you can have a DNA system? You can store all the information you'd ever need, records, photos, anything. It's simply another way of storing information. It had just been so slow up until 5 years ago, it wouldn't be thought to be practical. But it is. This is the first experiment showing what imagery you can hide in bacteria. This is the latest. It's a GIF file. It was actually programmed into the DNA of bacteria last year. The bacteria reproduced, and the offspring from the reproduction cycle would still produce this movie. Pretty cool. You can hide information in bacteria, and when the bacteria multiply, they can go into a spore form and last for a very long time. No one can scan you and find a bacteria. We don't have anything that can detect that. People, So if you want to be able to encode information, take pictures of information, create something in DNA and don't want it in your own body, it can be bacteria on some portion of your body, right? All they have to do is scrape it, let it grow in the Petri dish, and unpack the information. This is all available now. This isn't science fiction, but you can encode movies. Well, this is what the Chinese are doing with DNA. So in your own neck of the woods, you can begin inquiring. We are doing things with DNA as well. But the Chinese are fairly convinced that DNA encryption and encoding would be one tremendous challenge even for quantum computing. So this is where the race is right now, trying to merge quantum computing with what you'd call a wet hard drive with DNA. Merging DNA systems with quantum computing will be really quite an amazing and both lethal threat for that. The next thing I wanted to mention to you is memory. Can you play this one really quick? Hopefully you'll recognize this.
Hey, whoever you guys are, you're gonna have to show me some idea if you're gonna be in this. Hey, whoever you guys are, you're gonna have to... Hey, whoever you guys are, you're gonna...
Would you stop that? What? That thing is gonna give her brain cancer or something.
Hey, whoever you guys are, you're gonna...
So, what to do with memory? In medicine, we think of memory as a potentially harmful thing when people present with post-traumatic stress disorder. They can't stop thinking about the thing that's creating emotional distress. It's a very active development in the field to figure out, can we erase memory? Can we modify memory? Can we change memory? The short answer is yes. Several years ago, with the PAM Zeta data out of Duke University, this was the first time that anyone had ever demonstrated that if you wash an area of the brain called the hippocampus, it's an area of the brain that's crucial for forming short memories, spatial memories, and then facilitating the transfer from a short-term memory to something that's more permanent and stable over time, that he could train the mice to run the maze document the number of trials and errors, and then flood their hippocampus or expose it to this, and the memory would be completely gone. Meaning when the rats or the mice had to learn it over again, it was the same number of learning trials. And there was no trace of the memory left. Now the good news for us when we study rats and mice is we put electrodes and cannulae into their brain and can directly affect that area of the brain. If you wanted to poke your own hippocampus, you'd have to stick your finger through your eye and go right back in there. Sounds impossible to get to. Not if you program a cell to go there. So if you decide you wanted to program something that would selectively release PKM zeta after your meeting with someone, they probably would have no memory of it. That's what's happening in the rats, right? So the technical challenge right now is, how do we get a cell in there to do that in a human? I can assure you they're working on that in non-human primates right now. How many, what's the point specificity? Can we get it in there close enough to the hippocampus? Will those cells start reproducing in the next day, make enough of that stuff to wipe out a memory? Related to this, once you start thinking about memory, are chemicals that not only wipe out memory, but chemicals that enhance it. So if you want a better human camera, a better, an individual who can just go see and remember everything, that's the direction that the research in this lane is taking to help people with Alzheimer's. How to give them memory back. So what's being actively studied are the few people on the planet who have hypermnesia. In other words, they remember everything that's ever happened to them. We're actively trying to understand how to unlock that and unpack that and figure out why it is their memory does seem to record and they retain everything they've seen. They don't find it pleasant, and medicine would like to, people in medicine want to try and understand that so they can turn it into something beneficial for people who are losing memory. From a security and intelligence standpoint, it is a really unique opportunity to begin to discover, can you administer a drug that enhances human memory for a certain number of hours? Does it have to be permanent? So rather than carrying technical toys somewhere to try and record and collect information, your brain just remembers it, which doesn't give anybody anything really to detect. So that's one potential use for it, and that is one lane of research that's going on. That was just my picture to remind me of The Man Who Knew Too Much. If you remember the old Hitchcock film, essentially that's what he'd done. Remember, is memorize all the steps on how to make a bomb. He'd remembered the codes, he'd remembered everything. That research on hypermemory has gone more slowly than I thought in 2010. I thought by about 2015 there would be some progress. There hasn't been much yet in expanding memory very much. It seems to be a harder nut to crack than erasing memory. Erasing memory seems to be far easier. The last topic I wanted to review with you is memory. I don't know if you recognize any of the imagery up there, but I'll walk you through it. With memory, in the last 5 years, what's been demonstrated is that you can train a fruit fly around an aversive experience, and you can transfer that memory to the brain of another fruit fly by manipulating the rods. And it gives it a memory for something that it's never had before, and then it reacts to the stimulus in the same way as the animal who did have the aversive learning experience. It's been done in mice. I'll talk a little bit about what Beth Loftus and I have done to men and women going through SEER school and changing memory. And I put the last slide up because this is in flatworms, and this came out 2 years ago, that memory really is something beyond what we typically understand in flatworms. You can cut their head off, and their body still remembers stuff. So they're just beginning to uncode or decode where and how is memory stored in the body of this little creature, so we can translate that into memory in animals that look different than that little creature. It's evolved it for a very interesting reason. So this is in 2009 using light. They've transferred memory. You can turn things on and off using light in animals to activate the hippocampus, turn memory on and off. And so where are we with humans in creating false memories, giving them memories that they've never had? We've come a long way. My colleague is Beth Loftus, and this was her early work. It was called Lost in a Mall. And what she did is she asked a person to be in the study. You could be in her study if you had a sibling that was at least 5 years older than you. And she'd say, we're interested in your memory from when you were a kid. I've asked your older sibling, your older brother or sister, to give me 4 stories about you, and I want to know how much you remember. What people didn't know is that there were 4 different stories. One of them was fake, and she wanted to see how long it would take for them to adopt a false memory. The quick answer is, after 2 interview sessions, 30% of the subjects believed that they remembered the person who'd found them when they were lost at a mall, and actually argued with the researcher about whether or not the memory was true or not. And that's how I met her, and we decided to get together and run up to Brunswick to SEER School and try a memory experiment. This is our design. If you're not familiar with SEER, there's a classroom phase, there's an experiential phase. We were interested in sampling people when they were in isolation, when they were returning their gear, and at the end. And we tried a couple of different techniques. Group 1, there's no misinformation. We simply want to sample accuracy of human memory for their experience. And we told them at the beginning of SEER, we want you to be the best little human collector possible. We are going to quiz you about your memory. Don't let us trick you. We want to know what you remember. Group 2, we told them the same thing, but we lied. When they took their questionnaire at the end, we incorporated several techniques from false memory techniques, which are a little bit of leading questions to see whether or not we could create false memories. In the third group, we exposed them to an erroneous photograph of their interrogator. And in group 4, we used a video. So here's what we did. By exposing them to a photograph after they had been interrogated and placed in isolation stress, it could change them from this guy to this guy 48 hours later on who they would identify in the lineup. Their level of confidence was an 8 out of 10, that was the person they had met. We found out we could make them believe that there were guns, that there were knives, that there were caches of weapons, simply by altering the phrasing of a question or inserting something into a video. I'll give you an example. If we said, did your interrogator wear a weapon? If so, please describe it. We only got about a 2% endorsement of the presence of a weapon in the interrogation phase. If we said, when you were being interrogated by your interrogator and the guy with the weapon interrupted the interrogation, what did they argue about? We didn't care what the answer was. We'd ask another question. They'd say, describe the weapon worn by your interrogator. It jumped to 30%, would tell us the type of firearm that they had seen in the interrogation booth, which is a security violation, right? There weren't any. We got to record them. But with one question, we could do that. When you sample with a few more, you can actually increase the sample. So when we increased the stress at SEER, we found that instead of a 30% rate overall, we could create false memories in nearly everyone. That was in 900 people. So Beth and I were talking about that. We said, well, you can change memory. We know that. It's a way of understanding maybe why and how people have recovered memories of abuse that never happened. That's what her work has mainly been about. So she decided to do a study called Licked by Pluto. She decided she couldn't make Mickey Mouse a sex offender, but in her lab they thought Pluto was fair game. The short story is they were exposed to some misinformation about a man who had dressed in the Pluto outfit at Disney, and he'd been inappropriately rubbing his large fabric tongue on children, pleasurably and not pleasurably. There were 2 different conditions, and there was a neutral condition. If people adopted the false memory and their memory was for something negative, they did not want to buy the Pluto toy, right? When they went down their list, what they would not buy. She's done it with food. That was from her series with Alan Alda. She gave him a false memory that he'd been sick one time eating deviled eggs. And here they offer him one at the picnic on film, and you get the classic disgust wrinkle, and he said, no, I got sick one time eating them. It's not a true memory, it was planted. She's done it now with strawberries and ice cream, also done it with pickles, and has done it with alcohol. A study last year was that if you give college students the false memory that they were terribly hungover, they had a wicked hangover from drinking too much tequila, then when they're given free range options at the bar, like a week later, they decline it at twice the rate of everybody else. They go, nah, I got sick doing that. So think about it. If you change the past, you change human behavior. We are a case-based reasoning animal. When we think about what we're going to do, we think about the last time we did something, or what we heard about, or what we think it would have done. So to change human motivation, we don't have to persuade people. You can just change their memory. Think about the defensive and offensive capabilities of that. If you think about this from a defensive standpoint, you have the ability to change the memory of a person who's been debriefed in a safe house about the identities of who they met, the layout, because we've looked at altering memory for floor plans, for faces, for timing. If they're wrapped up by their intelligence service, they don't have anything to lie about, or what they remember is actually genuine, but it's wrong. That might be a defensive way of applying the technique. In medicine, people are arguing about whether or not you can use false memories to help people. Can I give you a false memory that leads you to stop smoking, or is it unethical? Because I can't tell you I gave you a false memory. I'd have to do it outside of your permission for your good. Most of us think it's probably unethical. In the society we live in, we think you probably should be an informed consumer, but it's a possibility that you can do. And when I think about this, I think about its relevance in this day and age when you start wondering what information is real and what information is trustworthy. When you start running into people and debriefing them, and you have sources who claim things, when you can learn how to create false memories, a person can be genuine and the information they remember is the old dangle idea. You can put information out that's simply not true. But in the current social media age, the ability to actually manage people's memories and change them is just enhanced compared to what it used to be. Now you can fix videos and pictures and expose people to audio and visual information, and we know that even if they know that's a possibility, people don't recognize when they adopt a false memory. It's a bit of a Trojan horse effect. You don't know that it's happened to you, and if you're smart and you have a good memory, you'll believe that happens to other people, but not you, because your memory is true. So it bypasses some critical reasoning on our part, and I think it's particularly effective. That's where the state of the art is right now for creating false memories in humans, is doing that verbally or by these manipulations with either what we say, what we show them, what we expose them to. But the chemical implanting of memories has now occurred in monkeys. So in trying to restore memory, there is probably, I would say in the next 2 years, we should see the science experiment come out that says a memory has actually been transferred or created and planted back into a human brain that wasn't done by a classic false memory technique. But I would anticipate that that's the direction the research is going. How do you rebuild memories in people who've had a TBI? Active research is going on about that on nanite reconstruction of brain cells and brain networks. And the idea in the mental health community is people lost part of their brain. We want to restore memory and brain function. Can we put the memories back in? So it's probably only science fiction for another 2 years, given the state of the art and the progress around that. And then, and the last thing I'll say, I didn't have any videos for it. I really wanted to show you one, but the French have published a very interesting paper, and it is this. While people were sleeping, they were able to train them and sample their knowledge in what they trained them in while they were asleep and while they were later awake and didn't know that they'd learned the information. So I'll say it again. In people who were asleep, they were able to tell what people knew around word recognition lists without ever waking the person up. They were also able to train new memory and information outside the person's awareness while they were asleep. Where that technology can go is some very interesting places. It would really raise, since I was in the lane of DS&T and we talked about deception and everybody was arguing about how to interrogate people, it raises an immediate question about whether or not you can sample information in people's brains outside of their awareness. The problem with a CAT scan and a PET scan and any technology is you have to have a willing subject. They do need to sit still. If people are asleep and you can begin to sample what their brain recognizes, it offers a number of opportunities at looking at guilty knowledge, brain recognition waveforms, and sampling some kinds of information. I don't know how soon it will be when you can link someone's brain to somebody else's while they're asleep, but I would imagine that that can't be far off. I'd probably ballpark it and say probably 5 years. If they have to do the brain implants, we'll know sooner, because I I can't see any other reason why DARPA got approval for 500 deep brain implants. I think the next step is going to be a hive brain. It's already been done in rats. You can link multiple brains, and as a hive, they solve problems much faster than the individual rat. So that technology's here. I'm assuming they'll link people who've given permission to link their brains to have a productive life, live in virtual reality, move robotic things. They can probably problem solve. So I think in the next few years, that's what we'll see, is brain-to-brain linking for problem solving to see if it makes it more efficient. But those are a couple of technologies I want to make you aware of. And then you can run away and think about their more direct applications. I tend to think of things from a medical perspective and from an intelligence and information perspective. But it's no longer really science fiction, and most of these fields have moved faster than I actually thought in 2010. The only one has been expanding memory that hasn't moved as fast. But I thought I would share that with you, and that's all I had to say. So thank you for your time. Yeah.